Bismillahirrahmanirrahim. Assalamu alaikum, dear students. I am Professor Dr. Shahzad Farooq. Today we are going to discuss gluconeogenesis. This is the continuity of our previous lecture. In previous lectures, we discussed the substrate and the main reactions of the gluconeogenesis and the biomedical importance of this pathway and the relationship between glycolysis and gluconeogenesis. Today we are going to discuss the non-equilibrium reactions of glycolysis which are reversed in gluconeogenesis by other enzymes. We discussed these reactions in previous lectures as well, but for the better understanding, we are again discussing these reactions. These non-equilibrium reactions of glycolysis are catalyzed by pyruvate kinase, phosphofructokinase 1 and glucokinase or hexokinase. Pyruvate kinase catalyze the conversion of phosphoenol pyruvate into the pyruvate, and phosphofructokinase 1 catalyze the conversion of fructose 6-phosphate into fructose 1,6-bisphosphate, and this glucokinase or hexokinase catalyze the phosphorylation of glucose into glucose 6-phosphate. These are the reactions of glycolysis. In the gluconeogenesis, the reversal of these reactions are not accomplished by these enzymes, but separate group of enzymes causes the reversal. For example, the conversion of pyruvate into phosphoenol pyruvate is catalyzed by the pyruvate carboxylase and phosphoenol pyruvate carboxykinase. This pyruvate carboxylase causes the carboxylation of pyruvate into oxaloacetate, and this phosphoenol pyruvate carboxykinase causes the decarboxylation and phosphorylation of this oxaloacetate into phosphoenol pyruvate. We will discuss this in coming slides. Then the other non-equilibrium reactions of glycolysis, which was catalyzed by the phosphofructokinase 1, was conversion of fructose 6-phosphate into fructose 1,6-bisphosphate. Its reversal, that is conversion of this fructose 1,6-bisphosphate, fructose 1,6-bisphosphate to fructose 6-phosphate is catalyzed by fructose 1,6-bisphosphatase. In glycolysis, the conversion of this fructose 6-phosphate into fructose 1,6-bisphosphate is catalyzed by the phosphofructokinase 1. And in gluconeogenesis, the conversion of this fructose 1,6-bisphosphate to fructose 6-phosphate is catalyzed by the fructose 1,6-bisphosphatase. The last non-equilibrium reactions of gluconeogenesis is the conversion of glucose 6-phosphate to glucose, which is catalyzed by the glucose 6-phosphatase in gluconeogenesis. On the other hand, the phosphorylation of glucose into glucose 6-phosphate in the glycolysis is catalyzed by glucokinase and hexokinase. We will see these reactions in coming slides. Now, how this pyruvate is converted into phosphoenol pyruvate. In glycolysis, this phosphoenol pyruvate is converted to pyruvate by the pyruvate kinase enzyme. But as you are watching, this is unidirectional reaction, so the same enzyme do not cause simple reversal of this pyruvate into phosphoenol pyruvate. This pyruvate is transported into the mitochondria where mitochondrial pyruvate carboxylase catalyze the carboxylation of pyruvate to oxaloacetate. This is an ATP requiring reaction in which the vitamin biotin is the coenzyme. This biotin binds carbon dioxide from bicarbonate as carboxybiotin prior to the addition of the carbon dioxide to pyruvate. The resultant oxaloacetate is reduced to malate, and this malate is then exported to the cytosol where it oxidizes back to oxaloacetate. Now, a second enzyme, phosphoenol pyruvate carboxykinase, catalyzes the decarboxylation and phosphorylation of oxaloacetate to the phosphoenol pyruvate using GTP as a phosphate donor. In liver and kidney, the reaction of succinate thiokinase in the citric acid cycle produces GTP as in other tissues. In liver and kidney, this reaction produces GTP, but on the other hand, other tissues produce ATP, and this GTP is used for the reaction of phosphoenol pyruvate carboxykinase. Thus, it providing a link between citric acid cycle activity and gluconeogenesis to prevent excessive removal of oxaloacetate for gluconeogenesis, which would impair citric acid cycle activity. Now, this phosphoenol pyruvate, which is formed from pyruvate by these group of two enzymes, is converted into 2-phosphoglycerate and then 3-phosphoglycerate and then 1,3-bisphosphoglycerate and then glyceraldehyde 3-phosphate, and ultimately it forms 1,6-bisphosphate. Here comes only the simple reversal of glycolysis reactions. But this fructose 1,6-bisphosphate converts to fructose 6-phosphate, and this is catalyzed by fructose 1,6-bisphosphatase. Its presence determines whether a tissue is capable of synthesizing glucose or glycogen, not only from pyruvate but also from triose phosphates. It is present in liver, kidney and skeletal muscle but is probably absent from the heart and smooth muscles. Here this enzyme causes the reversal of this or the conversion of this fructose 1,6 bisphosphate into the fructose 6 phosphate, and this fructose 6 phosphate is isomerized into the glucose 6 phosphate, and the conversion of glucose 6 phosphate to the glucose is catalyzed by the glucose 6 phosphatase. It is present in liver and kidney but absent from the muscle. So it cannot export glucose into the bloodstream due to deficiency of this enzyme. So in this reaction, glucose 6 phosphate is ultimately converted into glucose. Now, how the glucose is synthesized from different substrates like lactate, glycerol, propionate, and glucogenic amino acids. If we see how this glycerol, which is released from adipose tissue as a result of lipolysis of lipoprotein triacylglycerol in the fed state, it may be used for re-esterification of free fatty acid to triacylglycerol or may be a substrate for gluconeogenesis in the liver. In the fasting state, glycerol released from lipolysis of adipose tissue, triacylglycerol is used as a substrate for gluconeogenesis in the liver and kidney. Here it is phosphorylated by the glycerol kinase into glycerol 3 phosphate, and the phosphate is donated by the ATP. Then this glycerol 3 phosphate is reduced this glycerol 3 phosphate is oxidized into dihydroxyacetone phosphate, and this NAD is reduced to NADH, and the reaction is catalyzed by the glycerol 3 phosphate dehydrogenase. Again, I am repeating, this glycerol is phosphorylated into glycerol 3 phosphate, and the reaction is catalyzed by the glycerol kinase, and the phosphate group is donated by the ATP. And this glycerol 3 phosphate is converted into dihydroxyacetone phosphate, and this reaction is catalyzed by glycerol 3 phosphate dehydrogenase. As we know, dihydroxyacetone phosphate may isomerized into glyceraldehyde 3 phosphate, and the enzyme aldolase causes the formation of fructose 1,6 bisphosphate, and this fructose 1,6 bisphosphate is converted into fructose 6 phosphate by the enzyme fructose 1,6 bisphosphatase. And now this fructose 6 phosphate is isomerized to glucose 6 phosphate, and this is converted into glucose by the glucose 6 phosphatase. These two reactions we have already discussed in detail. So we can say glycerol is phosphorylated into glycerol 3 phosphate by the glycerol kinase, and the phosphate group is donated by ATP, and this glycerol 3 phosphate is oxidized into dihydroxyacetone phosphate, and this NAD is reduced, and the reaction is catalyzed by the glycerol 3-phosphate dehydrogenase. And this dihydroxyacetone phosphate and glyceraldehyde 3-phosphate forms the fructose 1,6-bisphosphate by the enzyme aldolase. And this fructose 1,6-bisphosphate is converted into fructose 6-phosphate by the fructose 1,6-bisphosphatase. And this fructose 6-phosphate is isomerized into the glucose 6-phosphate, which is ultimately converted into glucose by glucose 6-phosphatase. And by this way, glycerol is converted into glucose. And how the lactate produced in muscles and erythrocytes converted into glucose, we can see lactate is oxidized into pyruvate, and this NAD is reduced to NADH, and this pyruvate is transported into mitochondria, where it is converted into oxaloacetate by the carboxylation of this pyruvate in an energy dependent reaction catalyzed by the pyruvate carboxylase. Now this oxaloacetate is reduced to malate, which is exported to cytosol, where it is again reoxidized into oxaloacetate, and this oxaloacetate is decarboxylated in an energy dependent reactions catalyzed by the phosphoenol pyruvate carboxykinase. Here GTP causes the phosphorylation of this oxaloacetate, and we have this phosphoenol pyruvate. And this phosphoenol pyruvate is converted by simple reversal of glycolysis reactions of glycolysis up till fructose 1,6-bisphosphate, where it is converted to fructose 6-phosphate by fructose 1,6-bisphosphatase, and this fructose 6-phosphate produced by conversion of fructose 6-phosphate produced by the reactions catalyzed fructose 1,6-bisphosphatase is isomerized into glucose 6-phosphate, and this glucose 6-phosphate is ultimately converted to glucose by a reaction catalyzed by glucose 6-phosphatase. And by this way, lactate is converted into glucose. Now, how the glucogenic amino acids convert into glucose? As we have already discussed, glucogenic amino acids are those amino acids which on catabolism yield either pyruvate or one of the intermediate of TCA cycle. So, alanine, serine, glycine, cysteine and threonine forms a pyruvate, and asparagine and aspartic acid forms the oxaloacetate, and this glutamine, proline, arginine and histidine forms the alpha ketoglutarate, and this methionine, valine, isoleucine and threonine forms the succinyl-CoA, and this phenylalanine and tyrosine forms the fumarate, and they ultimately converted into malate, and this malate is exported and then it is converted into oxaloacetate, and finally it is converted into phosphoenol pyruvate, and as we have discussed, it will ultimately converted into glucose. Now, how the propionate is converted into succinyl-CoA? This propionate is converted into propionyl-CoA by an ATP-dependent reaction catalyzed by acyl-CoA synthetase, and this propionyl-CoA in an biotin-dependent enzyme propionyle-CoA carboxylase enzyme causes the carboxylation of this propionyl-CoA into D-methyl malonyl-CoA, and which is converted into L-methyl malonyl-CoA by the enzyme methyl malonyl-CoA resmase, and this L-methyl malonyl-CoA in an B12 dependent enzyme methyl malonyl-CoA mutase will convert into succinyl-CoA, which will enter the citric acid cycle. And in the citric acid cycle, here it enters, and this ultimately converted into malate, which is exported to the cytosol, converted into oxaloacetate, and finally into phosphoenol pyruvate, and which is ultimately converted into glucose, as we have already discussed in details how this phosphoenol pyruvate is converted into glucose. And dear student, this all was about gluconeogenesis. In the next lecture, we will discuss the regulation of gluconeogenesis. Thank you very much.